If you're in the right place, my name is Jeff Wedgwood. This is the evolution class. The books that I brought today, I brought this one as a reminder. This is basically Evolution, What the Fossils Say and Why It Matters by Prothero. It's an excellent book going through many, many different fossils to show how it supports the theory of evolution. The main thing we're going to be doing today is discussing the contents of this book called The Relics of Eden by Daniel J. Fairbanks. I brought this one because this book, it covers the discovery of Tiktaalik, which I went over last time, or a couple times ago, where the fish developed shoulders and a flat head and was able to come out of water. This is an excellent book, an easy read. This is the book that I think is best for this course, Why Evolution is True by Jerry Coyne. And this is The Making of the Fittest by Sean B. Carroll, which is also an easy read about evolution and how animals compete. Today, I want to go over the DNA basics that we did last time very briefly, and then I want to go into more advanced DNA information. And it's going to be very complex, so I'm going to go slowly, and I'm going to repeat myself to make sure that you get the ideas. First, I'm going to skip those guys. I'm just going to go straight to DNA. Remember that DNA is a double helix that has adenine, thymine, guanine, and cytosine, what are called nucleotide bases. We talk about them as A, T, G, and C. And there's what's called a backbone of sugars and phosphates, which is what makes up the backbone of the skeleton of the DNA. This image just shows DNA that's been opened so it can be duplicated for reproduction. Everybody clear on the A, T, G, and C? One of the things that's important is that A always pairs with T and G always pairs with C. And you see these little extra lines between the O and the NH2 and the NH and N there. All of those are what are called hydrogen bonds. They're fairly powerful bonds. They're bonds related to the electric charge, but they hold the DNA together. One of the points that I think Bill asked last time was, what's the difference between DNA and RNA? And DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So the only difference is each sugar group in DNA is missing an OH. That's all. But it makes an enormous difference, because in RNA, that makes the backbone less stable. And so that if you were to form a double helix of RNA, it wouldn't form a stable bonding as DNA does. So RNA is used for other things. Okay, I'm going to go over this again. The central dogma of molecular biology. DNA makes messenger RNA, makes proteins. Enzymes are required at each step of the way. So there has to be some proteins which are enzymes or physical structures like microtubules. But this is the simplest way to think about how things work in the DNA world. I'm going to skip this one. Okay. This is just a screen to show you that in DNA and in RNA, to make the proteins, it's read by 3 nucleotides at a time. And so this chart, which is in your book, you start with the first base, and since this is RNA, we have uracil instead of thymidine, so it's A, U, G, and C, just a little bit different, but this is from messenger RNA. So if you went U, C, G, that's proline. That will give you a proline to add to the protein. These are the amino acids that can get added to the protein. You notice there's a lot of redundancy. Like over here, if you have a U and a U, and you have any of these, any of those, you'll get leucine. So those can mutate. The third one can mutate and have no effect on the protein. It'll be a neutral mutation. Also, you notice that there is a start and what are called stop codes, which tell the enzyme where to start creating the messenger RNA on DNA and where to stop.
So that has to do with growth hormones? Or?
No, it has to do with the enzymes that attach to DNA to read it and then make the messenger RNA. So this is all inside the nucleus right now. Yes, sir?
No, that's okay.
Okay. Is everybody clear on this? How it's a complicated slide. You have to go 1, 2, 3, but once you get it, you'll see that the main thing I wanted to show was that there was a lot of redundancy which allows mutations to happen which are neutral, which is called genetic drift, and they're stopped in the start codons. Oops, went too fast. A few details. This is in your book as well. A lot of enzymes have to work together. A lot of errors occur anyway. More DNA errors occur during our lives. That's how cancer develops, for instance. And duplication of DNA and error in the gamete-forming cells, that is the testicles and the ovaries, those are the only important place because those are what contribute to the progeny's traits based on the DNA. So my basal cell cancer was created because of sunlight exposure to my skin, bad sunburns when I was a kid, changes in the DNA of my skin cells, and eventually it developed into what's called a basal cell cancer. That was removed, and I don't expect to have problems with it. But that shows DNA errors in non-gamete forming cells, like my skin, had no effect on natural selection. Is everybody clear on that?
So if there is errors on just some DNA in a human or animal, whatever, and it occurs, that's what caused, would that be the cause of something that
well, if a mutation, for instance, if a mutation changed the amino acid that was made from this to that, it would change the structure of the protein, and that would change how well it worked. It might change it for the worse, in which case that's going to be a negative natural selection. It might be for the better, in which case that animal is going to be able to reproduce more successfully. Yeah. You're absolutely right. Okay, let's see. Did I get all of those? Okay. So it's the gamete-forming cells that are important ones in terms of the contribution to the progenous traits. Okay. Now we're going to get into some complicated things, and as I said, I'm going to go slowly on this. DNA, the structure of DNA, imaginary. There's a start codon here, there's a stop codon here. I should have labeled those. But DNA is divided up into what are called exons and introns. The exons are all gray and the introns are all pale. What happens is DNA gets copied to messenger RNA, including those introns. But enzymes cut out all the introns. And the enzymes are very specific to where they'll attach and what they will cut. And they will basically paste back together the two ends of the RNA. So you have one long strand of messenger RNA. And that messenger RNA will be turned into protein when it goes to the ribosomes. Everybody got that on exons and introns? I'll tell you what the introns are in a minute. Okay. As I said, messenger RNA does not remain unaltered. It gets chopped up into little bits, and the ends are spliced together. There are these sliced out pieces represent useless DNA. I've already mentioned that. I'm sorry, this is a redundant slide. Notice also that proteins do not remain unaltered. You've got the DNA, which forms the messenger RNA. That gets chopped up and put together to form the final messenger RNA. That gets turned into a protein, and you'd think it'd be done, but you're not. Proteins often have either lipids, which are fats, or sugars added to them. And they often have other things happen to the structure so that it will form properly. Like a metal ion will be added to hemoglobin, for instance. The iron in hemoglobin will have to be placed in the right place, for instance. Not all proteins form properly. You may have heard of prions or prions. Creutzfeldt disease or mad cow disease in England. Those are caused by proteins which normally fold like this, but then they start to fold like that. And if they start to fold like that and they bump into another one of their buddies, it will fold like that. And so there's a cascading effect which has no enzymes involved, has no ribosomes or other cell organelles involved. It simply is a change in conformation, and that's what causes damage in the brain, called
Is that like
spongiform.
sickle cell anemia? Sickle cell?
Sickle cell is a genetic trait.
Does that have to do with the blood flow?
It has to do with the blood cells curling. I'm talking about the proteins.
Oh, the proteins,
It's the
sorry.
That's the same kind of idea. You've got something that should be a certain shape, and it changes shape, and now, for instance, in sickle cell, those sickle cells will block capillaries and cause pain.
Cause what?
Pain. Sickle cell crisis is extremely painful. I took care of a lot of patients with that, and it was really unfortunate. Okay, I want to mention chromosomal banding again. We talked about this before. Early on, before they did any work with DNA, they knew that there were chromosomes because they could see them as the cells divided, and they could make slides that would stain the chromosomes in certain ways, for instance, the banding here. The middle of the chromosome is called the centromere. That's the part which is pulled when you separate it after they've duplicated. At the end are the telomeres. The telomeres is a repeated length of a series of nucleotides in the DNA. And as you get older and as the cell divides, it doesn't duplicate as well to the very end. So each time a cell divides, the telomeres get clipped off. So you may start with 100, and then 90, and then 80, and so on. So a cell will basically get to a certain point where it can no longer reproduce, and it will send a signal to itself to kill itself. It's called apoptosis. There's a telomere at each end, as you can see, but as those are duplicated, they get shorter. This is just to review. I have a number of slides on chromosomal crossover. You've got this chromosome is the same as that one. This one is the same as that one. They get duplicated, but they cross over as they duplicate. And in the end, you end up with this extra bit here, 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 and here as crossover.
Excuse me,
Sure.
on the previous slide, what's the purpose of banding, or is that just used for identification?
It's just used for identification. My banding will be different than yours, for instance. So long before we had better tests, people would use that kind of thing for paternity cases or things like that.
So that's what they use in criminal
That was one of the things they
forensics.
did. I was not pleased with that particular slide in terms of crossover because it doesn't show the actual crossing over. This one does. This shows that basically the sections of DNA will have portions where they will have similar nucleotides. Those similar nucleotides will be attracted to each other. Remember the A and the T and the G and the C, they like each other, they like to be around each other. So things will get swapped, and in the end you can see that everything is different. This is another one that shows where just that one in the middle, just this guy did the crossover, but it shows the significant changes that go on in the progeny. And again, I liked this one because it has a family tree. Also, it has the Y chromosome, the little guy, and mitochondrial DNA, which we haven't talked about yet, but we will. But if you look at the top and then come down, you can see that some crossovers already occurred. Also note that this Y goes to here, goes to here. This Y doesn't go anywhere because this is female. So that Y chromosome is no longer in the gene pool. It's gone. Because this person only had girls. But you can see, not only do you get the crossover from grandparent to parent, but you get crossover from parent to child. So now it's even more complicated with, you know, dark stripes, dark light stripes, and so on. It can get very complicated.
So if the grandparents had all boys,
Mm-hmm.
and the boys had all girls,
Yeah.
then the next generation, there's still a chance for having a male.
Yes, but they won't have the Y because as soon as a father only has daughters, his Y chromosome isn't represented anywhere because the girls all have XX.
So where does the male come from then?
Well, but what I'm saying is you have to have a father who has a son in order for the Y chromosome to be passed on, because it only goes to males. And so you've got XX is females and XY is males. And this Y chromosome, if you have, this is the symbol for female, that's the symbol for male. If you have children, if they have only daughters, this guy's Y chromosome didn't go anywhere because his X chromosome went.
So the Y would have to come from the next generation.
It would, yeah. But what I'm getting at is this one is lost. This Y chromosome is lost. Unless this person has brothers who have sons, that chromosome will be gone from the entire genetic pool, and it won't be able to have any influence.
So, unless you have brothers, but cousins don't matter?
Not as much. They have some shared DNA, but the Y chromosome specifically, let's see. If you didn't have brothers, but you did have male cousins, yeah, it would still work. As long as there's a single male ancestor that goes from male to male to male to male, the Y chromosome will be preserved. But it's one of the interesting things because when they did research to try and find out approximately how old the Y chromosome is, they found that it's not as old as the evidence that they looked at in the mitochondria, which is passed only by females. This little circle here is passed only by females, so you'll notice light gray, light gray, light gray. That's because the mitochondria are in the egg. So when the egg is formed, it has the mitochondria, and each mitochondrion has a little circle of DNA that it uses in combination with the DNA in the nucleus to create energy out of sugars, for instance. But it's interesting here, we just talked about this Y chromosome disappeared, and now if we look here, this mitochondrial DNA disappeared. So you can only follow through the females for the little circle of mitochondrial DNA, and you can only follow the Ys through the males. Is that okay? Yes, Lenny.
What happens when, what is it, XXY or XYY?
Yeah, when you have multiples,
Yeah.
most of the time they're infertile.
Oh.
Yeah.
So most of the time, they can be passed
But
on.
I'm not sure how, I'm not aware that that's ever been passed out.
Friends, back where I lived before, they had in multiple units of a family, only males, only boys, one girl.
And she's the only one that will carry the mitochondrial DNA. And they are the only ones that will carry the Y chromosome from the parents. Isn't that fascinating? So there's this nice set. The Y chromosome you can check for male ancestry. The mitochondrial DNA you can check for female ancestry. And the estimate of age for the Y chromosome, I may be wrong, you can check your book syllabus, but I believe the Y chromosome has been aged at about 50,000 years, and the mitochondrial DNA has been aged about 70,000+ years. So the quote unquote, you've heard mitochondrial Eve, perhaps. That's completely made up. She's not Eve, and she wasn't the only one there. She's just the one whose mitochondrial DNA survived. Because as you go forward, it all depends on whether or not the progeny are male or female.
So if she was taken from Adam's rib, she would have a Y chromosome.
Yeah, that would be weird. I think there was a little fiddling there when that happened.
The reason why I become so interested in genetics is personal. Because I wonder why, when couples marry, you have to go for tests, you know, blood tests and stuff. Why, at this time, we don't so much, that couples are not tested genetically to see whether they're related, which happened to me after 1,500 years.
Yeah.
My husband and I met, we were like brother and sister genetically.
Oh my. That's a
The end
surprise.
up was bad, you know, the result was bad. But I wonder why this is not done, knowing so much.
I think it's because people know to avoid cousins
Who knew?
in marriage. I mean, they know that generally. But gosh,
I think
that's
they go
just
for genetic counseling if there's any problem.
if they
like
know of a problem.
So they know it,
palate
that's the
or
thing.
some mutant-
Now they know it, so there's genetic counseling, but then
Yeah.
they knew nothing about it.
You're right. You're right. Are you comfortable with this?
Sort of.
Do you rock it? Okay.
Well, I'm sorry.
Sure, go ahead.
In other words, are you saying that the Y chromosome goes back 50,000 years,
Can be dated.
80,000 years, and the X...
The mitochondrial DNA.
Mitochondrial goes back even further.
Even further.
And so in 50,000 years or 80,000 years ago, now we were 8 billion people, and 7 billion
Yeah.
people.
There was only, no, at this time, if you go back that time, they believe based on genetics, and I can't get into that because I haven't studied it, but based on the genetics, they say there was a bottleneck. Modern humans evolved, and then they went through an environmental change or a drought or a disaster or something, and it shrank to something like 1,000 people, which is a tiny thing to begin with.
1,000 people all over the world.
Yeah, over the entire world at that time.
And so 1,000 people means 50,000 or 80,000 or 150 X chromosome or Y chromosome.
I can't tell you when that bottleneck occurred, but that's one of the reasons that they can't go back any farther, is they think that there was this bottleneck, that the
Okay.
population was genetically fairly
And so
homogeneous.
That wasn't it.
in that bottleneck, say,
Noah's Ark.
I am related to a man 80,000 years ago as my great 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 great
Dot, dot,
great
dot.
great great
Yes.
great 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 grandfather.
Correct. Okay, we're going to talk about something which is a little confusing. So I'm going to go slowly with this, and I want to make sure that it makes sense. Barbara McClintock was a brilliant researcher who published articles on maize, on corn, and she got interested in how is it that you get the corn, the type that you see around Halloween, with the different colors on the different kernels. And she discovered, or she wrote, an article saying, you know, I can't prove this, but I think this is the way it works. And she talked about pieces of DNA that would move from place to place. And I didn't put a slide in there, and I apologize for that, but let me draw it on the board. what she said was, if you have a strand of DNA, and I'm stretching out the helix, if it has repeats of the same code, A, or ATG, 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 and you have the same set of duplicated codes at the end of a segment of DNA, those will wrap around, and what will happen is you'll get a loop. And that loop is primed to be cut off here, which is fascinating. She then said, okay, now this free piece of DNA can go somewhere else where there's another strand of DNA, and it can insert itself here. I'll get that blue. It's hard to see around. So what she was saying, I think these things are moving to a place which changes the activation. That is, whether or not an enzyme is going to be active in the corn kernels. So each kernel had a possibility of having this happen so that the kernel might be clear, red, brown, and so on. She called these transposons because they transpose, that is, they transport themselves from one place in the DNA to another. This can happen on the same chromosome, it can happen on a different chromosome. They are very common. We have thousands and thousands, if not millions of them. Some of them are called endogenous retroviruses, and I'll tell you what that means. And it's interesting because the same transposons in humans and chimps are found in exactly the same spot in the DNA, which points to a common ancestor. Now, I want to make that point really clear, so I'm going to come over here again. Let me get the light sequence. Okay, this is humans, and this is chimpanzees. When we say they have the same transposons in the same places, what we mean is, if you look at their DNA, they will have these little bits of DNA that have been inserted in exactly the same place. And if they didn't come from a common ancestor, it would be very difficult to form a hypothesis to account for this. Because this is useless, remember? This is the stuff that gets cut out. It's the introns that get cut out. So the only logical reason that chimps and humans have the same transposons in exactly the same locations with exactly the same codes is to have a common ancestor. Does everybody follow that? You can also use that information to look at relatedness, which we'll do in just a moment. Can you see this with the light on?
Yeah.
I should have asked.
Leave the light on.
Why?
I'm sorry.
The same code, you have the same ancestor. Why?
I don't understand.
Okay.
The reason is that probably happened in a common ancestor to humans and chimps, and it happened before humans developed and chimps split off. Humans and chimps split. So those places that I had marked in red must have existed already before humans and chimps split. Fascinating stuff, I think. Okay, let me take a step back. Endogenous retroviruses. You've heard of HIV, right? Okay. What does HIV do? HIV injects itself into a cell, it hijacks the ribosomes, creates proteins which are retro DNA transcriptases. Oh, never mind. They're just retro. What happens is this creates an enzyme, and that allows it to insert itself into the DNA of the cell. This is one of the reasons HIV is so hard to treat, because now this gets duplicated, and HIV DNA is created, and RNA is created, just like a messenger RNA, and it gets packaged and gets sent out of the cell. But many of these, there are many other retroviruses besides HIV, and they all entered our DNA using the same mechanism. And we have thousands or millions of them. Right now, most of them are inactive because they've been mutated. And one of the things that's interesting to me is things called fossil genes, as well as immortal DNA. Some DNA, its purpose in the cell, is the same all the way from an amoeba to a human. We all metabolize sugar, we all metabolize fat, we all create cell membrane, we all create nuclei. So we have enzymes which are very common to all of us. For instance, on the order of 70% of our DNA is the same in plants. Because all of those things that are at the very basic mechanism of just how does a cell stay alive are the same. And some of these, some of the genes are so important that even a single change in a nucleotide will make the function of the enzyme or the protein fail. So the DNA for that particular thing is exactly the same in everything you test that's a living thing. And they're called fossil genes. They're no longer expressed, I'm sorry, those are called immortal genes. Those ones where they're completely unchanged and you can see them all the way through the animal kingdom and the plant kingdom. Fossil genes are genes which have undergone a change in their activation site. So they're no longer used. The start codon that I mentioned on the in that slide with all the amino acids, the start signal may be gone, which means that you now have a gene which no longer functions, and if the animal can still survive, that gene will sit there, and over time it will accumulate mutations, and the mutations occur at a relatively stable rate in terms of, depending on where you look. So you can use these number of mutations in the fossil genes to guesstimate how old that gene is, and that's one of the things I did with the Y and the mitochondrial DNA. So if we look, two animals with the same fossil genes, that is, unexpressed genes, can give us an indication of how long ago the common ancestor was. So if you have, for instance, a fossil gene like this, and it starts to accumulate mutations, because it's not a gene, because it's a fossil, those mutations can occur without having any effect on the cell or on the animal being able to reproduce. So you can just look over time and count the number of mutations in fossil genes between, say, chimps and humans, and you can say, oh, well, they probably had a common ancestor about this long ago. That's where I got the numbers for the Y and mitochondria.
I have a question. Maybe it's a little bit off the subject, but
Sure.
a few years ago, National Geographic was doing research, and would order a packet. I took my DNA, I swap it, send it in, and they were able to trace where my family history came from, from the maternal side.
Right.
And it was interesting, they sent me a little disc, I put it in my computer and showed where it started from,
Yeah.
and where it spread out.
We're going to be talking about the geographic study when I come back from my vacation.
Okay.
And I have to write up a whole new handout for that. When you do DNA comparisons and you're just looking for overall similarity, you might use the fossil genes, you might use the entire genome. It depends on what you're going to look for. But if you use the entire genome, these are the relationships between these animals: humans, chimps, whales, dolphins, hippos, and camels. And you can see that humans and chimps share 98% of our DNA. Alpacas and camels share 99, and the lowest is around 79. So humans have 79% of the same DNA as all of those other mammals. Now the thing, you can take this chart and you can say, okay, who's related to who? Well, the most obvious thing is there's 0.99s here. So dolphins are really related to whales, and alpacas are really closely related to camels. So the first thing you do is you just make a little graph. At the bottom, you put the percentage of the similarity. This is looking at growth hormone. So you say, okay, humans and chimpanzees are 98%. Notice it's a little further out than the dolphins and whales and the camels and alpacas, which are 99. You then go back and you say, okay, what's the next closest thing? 97, 97, a bunch of 96s. And so you plot those. And so you can see that there's, the whales and dolphins are related to the hippopotamus, which is fascinating. I showed you the